Welcome to the first video on the cloning unit that we're doing in class. In this unit we're going to be doing something called cloning the green fluorescent protein, or GFP. In this video I'd like to introduce to you the idea of cloning, what that means in the context of what we're doing in class, and also give you a little bit of a history of how GFP was originally cloned. What is cloning? I think a lot of you probably, when you hear the word cloning, think of making a copy of an organism. For example, making a genetic copy of yourself so that there are two of you running around. You might be thinking of Dolly, for example, who was it was a sheep that was the first mammal to be cloned using a certain technique that we're not going to discuss today, but that was never used before in mammals. And so she became the most famous sheep in the world because she was a clone of another sheep. And while that is true, that's a good use of the word clone, it's not quite the way we're going to use it in this class. So when we talk about making a mirror image of yourself, or Dolly, or an animal clone, what we're talking about is animal cloning. And that's not what we're going to do in this lab, but I wanted to just make sure that you know these terms. Animal cloning, this is when we make a genetically identical organism, animal, and I want you to distinguish that from what we're going to do in class, which is molecular cloning. When we talk about molecular cloning like we're doing today in this unit, we are actually making copies of something, but in molecular cloning it may just be a gene that you're copying. And so often when you say, I'm going to go clone a gene, what you really mean is I'm going to make a copy of a gene, or I'm going to isolate a gene from a genome. So you've got a chromosome here, and you've got a particular gene on that chromosome. What you want to do is isolate that gene from the rest of the chromosome somehow and make copies of it, and then put it in some sort of carrying case so that you can replicate it and keep that gene in the lab to use for other applications. So in cloning, when we say molecular cloning, really what we're talking about is copying a gene, copying and isolating a gene. And so that's what we're going to be doing with GFP, is copying and isolating the GFP gene. So here's a more formal definition of molecular cloning. It's a set of experimental methods in molecular biology that are used to assemble recombinant DNA molecules and to direct their replication within host organisms. So you probably, of all the words in here, you probably are least familiar with that one and maybe what this means. So let me explain what we're doing then. So in molecular biology, we're going to take some sort of DNA fragment that we have obtained either from some source. And what we're going to do is make copies of that gene and put it into some sort of carrying case that we refer to as a vector. So here we have this DNA fragment and this carrying case, and we're going to combine these two things together. And so this carrying case, this vector, is a circular piece of DNA that we refer to commonly as a plasmid. And basically what we want to do is put our gene into that plasmid. And why would you want to do that? Well, these vectors, or these plasmids, are a very convenient way of holding a gene or holding a piece of DNA and producing a lot of it. Of course, in molecular biology, if you are wanting to learn how a gene works, you want to be able to do multiple experiments with it. And so you need to produce a lot of that gene so that you can do a lot of experiments with it. So if you put it in a vector, that allows you to amplify that gene and make lots of copies of it, or clone it. And so in this case, there are two ways to do this, to put it into that carrying case. It may be that your DNA fragment is linear, and so this gene you have, it comes in a linear form, like our DNA, humans' DNA is in linear form, right? Bacterial DNA is in circular form, and so you need to combine a linear fragment with a circular fragment. And so what you have to do is cut open that circular fragment to make it linear, and then you can paste in your gene of interest into that plasmid. It may also be that your DNA of interest is already in some sort of vector and you want to transfer it to another vector, so from vector A to vector B. So in this case, you would cut it out from vector A, and then you'd cut open vector B, and then you'd paste in that gene into the space created in vector B. And then you ligate these fragments together to create a new piece of DNA, circular piece of DNA. Then you can transform that piece of DNA into bacteria like E. coli, and those E. coli can take that bacteria and they can take that plasmid and replicate it, make many copies of it. Then you can collect it and put it in a tube and put it in your freezer, and you've got a lot of copies of that gene. And notice that that gene is isolated from the rest of the genome. We've done other things to isolate it from the rest of the genome, and so now you just have a copy of that piece of DNA, that gene, or whatever it is that you're copying. It might not necessarily be a gene. There might be other things that you're trying to copy and work with, other pieces of DNA. But in the end, you've got E. coli that are making many, many copies of that piece of DNA. So this is molecular cloning. So in a way, we are making copies of something. We're just not necessarily making genetically identical organisms. Although, if you think about it, when we grow E. coli that have this piece of DNA inside of them, then all of the E. coli that have that piece of DNA, if they arose from the same original E. coli cell, would all be clones of each other. But we're not really talking, when we say we're going to clone GFP, we don't necessarily mean that, we don't mean that we're going to grow multiple copies of the bacteria that are all clones of each other. What we mean is we're copying the gene GFP. There are lots of reasons that we might want to clone a gene or a piece of DNA. There's a lot of things we can learn about working with DNA and genes in cells, in animals. We can even use this genetic engineering or this cloning to create new types of organisms that have traits that they didn't have before. When we clone, we're basically recombining DNA from different sources. So recombinant DNA means that you have DNA from one source combined with DNA from another source, and you put it into a new organism that has never had that DNA in it before, and now that organism, we would say, contains recombinant DNA, DNA that's being recombined in new ways. And so you can make organisms that are different than anything that's ever existed on Earth before. You can make E. coli that have a jellyfish gene in them. E. coli have never had a jellyfish gene in them in all of history until now, when we do this genetic engineering and clone this piece of GFP, this gene GFP into E. coli. A lot of genetic engineering has been done on foods that we eat. And I just wanted to show you an example of a genetically modified organism, GMO, that has been made using molecular cloning. It's called golden rice. It's over there on the right. So golden rice is a genetically modified organism that was created using molecular cloning. A gene, or I think actually a couple genes, that are involved in beta-carotene synthesis, which is an essential vitamin that is not found in white rice. The genes to make that vitamin were cloned into the genome of white rice, so that now white rice produces beta-carotene, whereas it didn't before. And so molecular cloning has been used to produce a lot of different kinds of genetically modified organisms that we don't have time to talk about, but that have greatly, in many ways, greatly changed the way we grow our food, the way we think about food, the way we eat food, and of course has also created a lot of controversy. But genetic engineering or cloning or using recombinant DNA is not just used to create genetically modified organisms. Most of its use, the vast majority of its use, is in doing science, in figuring out how cells and our bodies work. When we can take genes and isolate them, then we can look under very controlled conditions at how these particular genes work in cells. We can take these genes and modify them and see how that affects cells, how it affects organisms. We can create mice and flies and worms that are all genetically modified and see how genetic modifications change the way these organisms develop and live. So we can learn a lot about how genes work, how different genes affect our health and how we live. And so the vast majority of genetic engineering has been done in the lab to study genes and how they work. So I wanted to talk to you about GFP because this is the one we're going to be using in the lab. GFP is a protein that is produced by a jellyfish called Aquaria victoria. It lives, I think, in the Atlantic Ocean. It's actually fairly rare, but these jellyfish have two kind of neat things about them in terms of the light that they give off. The one thing so the first thing they have is bioluminescence. So they have a protein in them, bioluminescence. They have a protein in them called aquarin that I haven't shown here. It can actually produce light on its own. It can be bioluminescent. And then there is fluorescence from the GFP. Now, fluorescence is different from bioluminescence in that fluorescence requires an input of light in order to get an output of light. And so this is a picture of GFP here. And so blue light comes into this protein and green light is emitted. And so we have an absorbance of blue light and an emission of green light. This is a protein that these jellyfish produce. I'm not sure why they produce it. It probably has something to do with them finding other jellyfish of the same kind. But I want to take a look at this protein a little bit and show you a little bit of its structure. I hope that one of the first things you see when you look at this GFP is that it has a barrel-shaped structure. So this protein, it's a single protein, it's a single polypeptide that when it folds forms this barrel structure, and in the middle of that barrel is a group of 3 amino acids that form what we call the fluorophore. And these 3 amino acids that are in the middle of this barrel are oriented in such a way that when blue light comes in and strikes them, they absorb that blue light and then emit it in the form of green light. And this barrel structure that is around them, that's also made of amino acids, that's also part of the same polypeptide chain, it's necessary for that fluorophore to be able to produce that fluorescence. GFP is found on one of the chromosomes. I don't know how many chromosomes a jellyfish has, but it is a gene found on one of those chromosomes. And I wanted to go through with you and explain in a brief way, a lot of times, we don't do this in lab, we're not going to do this in lab, but how did we get GFP, the gene, from jellyfish? It's hard to overstate how important GFP has been in the progress of science. In fact, some of the early people who cloned GFP and then figured out how to utilize it practically in the lab have won Nobel Prizes. It's an extremely important protein, and the reason for that is that it has been used for many, many years now as a marker. So what do I mean when I say a marker? What I mean is that it has been used for many years to be able to look at what's happening inside cells that are living. Before GFP came along, it was very hard to know what proteins were doing inside of cells, because the only way we could look at these different structures in the cells were to fix the cells, which killed them, and then look at them under a microscope with different stains. And while that told us what was we could basically figure out what was happening in cells with those fixed cells with stains, there was a lot we couldn't figure out. For example, how do cancer cells move around inside a body? If we have to just fix and kill the cells every time we want to watch what they're doing, then we won't be able to watch dynamically how they're behaving. And so GFP has been used to mark different parts of the cell so that we can watch what's happening in real time in living cells. GFP expressed in cells is non-toxic, and so it doesn't affect the health of the cells, and it doesn't need anything to fluoresce. All we have to do is shine blue light on it, and it automatically fluoresces. So if we have a living cell and we have GFP in there and we shine a blue light on it, we can see the GFP and we can watch what's happening in the cell. So one thing that you might want to do is say, okay, I am really interested in what this protein does in the cell. And so using molecular cloning, you can tag or mark, thus the word marker, or tag GFP to that protein molecularly, so that when that protein is expressed, GFP is expressed also. And then you can express that in cells, and as the cells do their thing, you can put them in a dish, or you can leave them in the animal and image the cells in an intact animal, and you can watch what this protein does, because all you have to do is shine blue light. And wherever you see green, that means that's where the protein is, because the protein and the GFP are covalently linked together. And so this idea of using GFP as a marker has been invaluable in figuring out how the cells work. It has taken us leaps and bounds ahead of where we were when we were just fixing cells and looking at them under the microscope. Anyway, I won't go on. It's hard to state the whole scope of how GFP has been used in science for the past, 30 or 40 years now. But I just want you to know it's been very important in learning how our bodies work and how our cells work. So how did we originally clone GFP? How did we originally get it out of the jellyfish to use in the lab? I just want to briefly explain how that happens, because I think it's kind of a mystery. It's kind of a black box that we never have time to explain in class, and so I wanted to take a little time to explain it right now. So here we have our jellyfish, and it has the gene for GFP in all of its cells, and in some of its cells it actually expresses the GFP gene. Now, how does it express the GFP gene? Well, here's the chromosome, right? So let's say this is the chromosome. And on this chromosome, there's a gene for GFP. And when the jellyfish expresses GFP, it makes an mRNA for GFP through transcription. And then through translation, that mRNA is read by the ribosome to produce the protein GFP. So transcription makes the mRNA, translation makes the protein. That's how the jellyfish normally expresses it. So how do we get the GFP gene in our lab? Well, what we're going to do is we're going to take the mRNA of jellyfish and we're going to create something called a cDNA library. Now what we're going to do, our whole goal here is to produce, let's just put over here, our goal is to get an isolated GFP gene. So the GFP gene is currently in the chromosome here, surrounded by lots of other genetic material, but we want to isolate out the GFP alone. Well, when the jellyfish, again, when it transcribes the DNA into mRNA, then what we have here is an isolated GFP mRNA. Okay, now what we want to do is change that back into DNA, because what we want at the end, again, is a GFP gene that's made of DNA so that we can use it in the lab. So what we're going to do is take this mRNA and make a cDNA out of it. And what is that cDNA? Well, cDNA stands for complementary DNA. Now, how is this produced? So mRNA is a single-stranded molecule, right? And DNA is double-stranded. Normally, we don't produce DNA from mRNA, but there is an enzyme that can do this, and it's called reverse transcriptase. And it is a viral gene that we can take and use to our advantage to create DNA from mRNA. So what we're going to do is take a jellyfish, we'll chop it all up, blend up its tissues, and we'll collect the mRNA from those tissues. And then we'll apply reverse transcriptase, this enzyme that will convert all of the mRNA from this jellyfish into cDNA. And then through a further process, so initially that cDNA will be single-stranded, but then through a further process we'll produce double-stranded DNA. Okay, so let me do this in a different color here. So we've got mRNA here, and then reverse transcriptase comes in, and it will produce a complementary copy of the mRNA that is made of DNA. So I'm going to make my DNA blue here, actually. So let's just think of DNA as blue, all right? So here's the DNA. That's blue. Okay, so the reverse transcriptase makes this cDNA copy. And then we're going to use a regular DNA polymerase to produce a double-stranded cDNA. Now this cDNA is the GFP gene. It's just been reverse transcribed from the mRNA. Okay, so we've got this jellyfish, we've blended up its tissue, we've produced cDNA from the mRNA using reverse transcriptase. But I want you to think about these jellyfish. They don't just express GFP, they have lots of different genes that they're going to express. So not only will you have, when you do this process then, you can't specifically produce just GFP cDNA. You're going to be producing cDNA for a lot of different types of genes, all of which were originally mRNAs inside the jellyfish. And so in your tube, you'll have the cDNA for GFP, but you'll also have the cDNAs for all the other proteins that were being expressed in the jellyfish. And so this is why we call that a cDNA library, because it contains all the cDNAs that jellyfish had. So what do we do next? How do we isolate the GFP gene alone? Well, what we're going to do is take all of these cDNAs and package them. We're going to use something called a phage, which is a virus. And what we're going to do is do this little trick where we're going to put each cDNA into a different phage. So this will go into phage 1, this will go into phage 2, this will go into phage 3. And the way this is done is through molecular cloning. We're actually going to do a cloning procedure that allows us to package a single cDNA molecule into a phage, which is, again, a virus. And so at the end of this process, we will have a phage that contains each of the cDNAs that that jellyfish originally had. Or each of the cDNAs that we produced from the mRNAs that the jellyfish had. All right, so phage 1 will have the GFP, phage 2 will have this other gene, phage 3 will have this other gene. And I'm not going to go through all the details of how that's done. There's a really simple technique where you can insert this GFP cDNA and all these cDNAs into a plasmid, and you can, so this is a plasmid, and this plasmid has viral DNA on it. And so when you insert your cDNA into the middle of that viral DNA and then put it into certain cells, bacteria, the virus is actually produced inside the bacteria. So here's a bacterial cell, and we actually produce the virus inside. And the virus will pack, it will be the gene that we put, all the cDNAs that we put into that plasmid will be packaged into these different phages. And then we can collect these different phages, and now what we have is a phage library. We can think of it as a library of jellyfish genes. Now what we want to do is we want to figure out which one of these has our GFP gene, because we won't be able to know, just, viruses are too small to look at, and even if we could look at them, we wouldn't know which one has the GFP gene. So we've got to have some way of detecting which phage has the GFP gene, and then we can grow those phages and collect the GFP gene out of them. So the next step we're going to do is take these phages and put them onto E. coli. Now, what we're going to do, we have this plate here that's called an agar plate, and it's going to be basically carpeted with E. coli. They call it a lawn of E. coli. So think of this is a dish with a gel in it that E. coli can grow on, and then on the surface of that gel we've basically coated it with E. coli. Now phage infect E. coli, and when they infect E. coli, they burst open the E. coli. And when they do, the E. coli turn from an opaque white color to a clear color. And so if you have E. coli that have been damaged by phage, they will create these little clear spots on the dish called plaques. And a plaque is a region of the plate that has been cleared by one phage. So there are 5 plaques on this dish that I've drawn. So that would mean that there were 5 different phages that infected these E. coli. Hopefully one of those phages, so how did the plaque form? So the phage infects one of the E. coli cells. Inside that E. coli cell, it produces multiple clones of itself, and then it blows up that E. coli cell, and all those phages that are released infect the surrounding E. coli cells, and those all get infected, and they produce more phage, and then they burst open and release the phage to surrounding E. coli, and so this plaque starts out really small, and then it gets bigger and bigger, and pretty soon it forms a circle that's big enough for you to visualize and see. It's a clear spot on your dish, while the rest of the dish is kind of an opaque white. And so this plaque, this circle, this clear area, contains a bunch of phage that may or may not have your GFP gene. So we want to find out which one does, and so what we're going to do is we're going to pull some of this phage off and test each one of these plaques with a probe that we know will detect GFP. So what we're going to do is take a piece of paper that's like a filter paper, and we're going to put it on top of this dish, And when we put it on top of this dish, some of those phages will stick to that paper, and they're going to stick to that paper in the same orientation that they were on the dish. So that then when we take this piece of paper over here to do our experiment with, they will be in the same orientation as they were over on the dish. Now what we're going to do with this piece of paper is we're going to add a probe to it. So when we originally started working with GFP, we got a good idea of what the amino acid sequence was for GFP by doing some techniques using different enzymes and different types of ways of looking at the protein that I don't have time to go into now, and I don't know that I could even explain. But we've got a good idea. We have, over the years, figured out what we think the amino acid sequence of GFP is. And based on that, and knowing what the translation language is for DNA, we can back translate from what these amino acids are. Okay, here's some amino acid symbols. I don't know if these are the exact amino acids in GFP, but we know what the basic amino acid sequences of GFP, and then we can backtrack and say, okay, the possible DNA sequences for tyrosine are ATC, ATG, and the possible codon sequence that encodes for R, which is, I think, arginine, is GTC or GTG. Okay, and then for alanine, it's TTC or TTG. And so we can say, okay, here is our best guess at what the DNA sequence of GFP is. These phages, one of them at least, has the exact DNA sequence of GFP. What we're going to do is try to find that one. And what we're going to do is create probes that contain, we're going to artificially create small pieces of DNA that have either this sequence, or this sequence, or this sequence, or this sequence, or this sequence. We're going to create a lot of different probes that have different combinations of this sequence here, and one of them is going to be the correct sequence for GFP. And so one of these probes will actually bind directly to the GFP gene because it'll be complementary to the GFP gene. It'll be perfectly complementary. So what we can do is create a bunch of these probes and then tag them all with some sort of radioactive molecule so that when we apply them to this paper, they will anneal, if they are complementary to the GFP gene, they will anneal or stick to that part of the paper where the GFP gene is found. Then what we can do is look for radioactivity. And let's say that we look at the radioactivity and we see radioactivity right here. That means that these probes that were complementary to GFP stuck to this plaque. That means this plaque has DNA that is complementary to these probes. And since these probes were designed to be complementary to GFP, we can assume that this plaque here has the GFP gene in it. Once we figure that out, we can come back over here to this dish and we can scrape some of that plaque off and grow those phages in even more bacteria and produce many, many more of them, clones of them. And then we can collect, once we've got a bunch of phage, and they're all genetically identical because they all came from the same plaque, then we can isolate that DNA, and that DNA that we isolate will have the GFP gene in it. And that's how we isolate it from all of the other genes that were in that cDNA library, is by using these probes that will seek out and find GFP alone. Once we have that GFP gene isolated, we can then take that GFP gene and cut it out of that plasmid and put it in to some other plasmid and create these clones. And so then we can use that GFP. So that's originally, this was done back in the 90s, early 90s, by a man named Prasher, and I believe he won the Nobel Prize too. He was the first to clone GFP from jellyfish. And it was like, once he did this, jellyfish. Once he did this, the horses were out of the gates. For some reason, my pen is not writing anymore, but that says jellyfish. Once he did this, the horse was out of the gate, and we were off and running and had an explosion of knowledge of cell biology and human health because of all the possibilities that having GFP and being able to use it in different applications gave us. So I hope that was interesting to see how we originally went from a jellyfish to having GFP isolated by itself. And I hope that was helpful. Be sure to ask any clarification questions in class. But I think this video hopefully will save us a lot of time and prevent me from having to spend a lot of time in class explaining. And instead we can get our hands dirty and start doing the actual techniques.